We've all seen those amazing pictures of cells, the ones in textbooks.
Right.
But can you imagine actually seeing them in action?
Oh, absolutely.
Moving around,
Yeah.
dividing, responding to their environment.
It's incredible.
Like a whole hidden world coming to life.
It really is. And it's all thanks to this revolution in microscopy.
Oh, wait.
It's called live cell imaging.
Okay.
For centuries, we were stuck with just these static snapshots of cells
Right.
frozen in time,
Right.
But now we can actually observe these dynamic processes as they happen.
Wow.
It's giving us a whole new understanding of how cells function.
So is it like the difference between a still photograph and like a breathtaking documentary?
Exactly. And one of the key techniques that makes this possible is time-lapse microscopy.
Time-lapse microscopy.
Yeah. Imagine taking a series of pictures over time and then stitching them together to make a video.
Oh, cool.
But instead of like a blooming flower, we're watching cellular processes unfold, at high speed.
That's incredible.
We can see how cells migrate in response to signals.
Okay.
The whole process of cell division, even programmed cell death, all in this dynamic sequence.
Wow. So it's like stop motion animation,
Yeah.
but with real life cellular drama.
Right.
That's so cool.
It is.
So what sorts of things are researchers learning from these cellular movies?
Well, one area where it's been really impactful is studying cell migration.
Cell migration, okay.
So that's basically how cells move around,
Yeah.
and it's essential for everything from wound healing to immune response.
Yeah, I can imagine that being pretty important.
It is.
But how does actually, like, watching it unfold in real time, how does that change our understanding?
Well, for one thing, we can see how cells respond to these chemical signals in their environment. So they can actually sense gradients of these signals
Right.
and then move towards or away from them.
Wow.
It's like they're following a map, only we can't see it.
So it's not just random movement. They're actually, like, navigating.
Exactly. They're navigating their surroundings.
Yeah. And with time-lapse microscopy, we can track their entire journey and observe how they interact with other cells and structures along the way.
Wow.
It's helping us unravel these complex mechanisms that guide cell movement,
Okay.
which has huge implications for understanding development, disease, even the spread of cancer.
So we're literally watching cells make life or death decisions in real time.
Pretty much.
That's incredible.
It is.
But they're so tiny.
Right.
How do we even see what's going on inside of them?
Ah, well, that's where fluorescence microscopy comes in.
Fluorescence microscopy.
Think of it as like painting cells with light.
Oh, okay.
We use these special fluorescent tags that bind to specific molecules or structures within the cell. And then when we shine a particular type of light on them, they glow.
So it's like highlighting different parts of the cell with different colors.
Exactly.
So you can see them against the background.
Yeah, make them stand out.
Okay.
There are two main types of these tags.
First, we have fluorescent proteins, like the famous green fluorescent protein, or GFP.
Right, GFP.
We can genetically engineer cells to produce these proteins, and they act like these little tiny beacons, allowing us to track the movement and the interactions of specific molecules within the cell.
So we're essentially turning the cells into their own little light shows.
Yes, you got
That's
it.
pretty neat.
It is.
Yeah.
And then we have fluorescent dyes,
Okay.
which can be used to stain specific structures within the cell, like the cell membrane or the nucleus.
Right.
This lets us visualize the architecture and the organization of the cell in just stunning detail.
Okay, so we've got time-lapse microscopy to capture the movement,
Right.
fluorescence microscopy to light up the scene.
Yeah.
But I have a feeling we're just scratching the surface here.
Oh, yeah.
What other tools are researchers using to delve even deeper into this cellular world?
Well, you're absolutely right. There's a whole suite of advanced techniques that take live cell imaging to another level. For example, confocal microscopy gives us these incredibly sharp three-dimensional images
Oh.
by using a laser to scan the sample in these thin layers.
So it's like a microscopic CAD scan,
Exactly.
revealing the intricate structures inside the cell.
That's a great way to put it.
Very cool.
It is.
So what other cool techniques are we using to spy on these cells?
Another fascinating one is called TIRF microscopy.
TIRF microscopy.
Yeah, it stands for Total Internal Reflection Fluorescence Microscopy.
TIRF. That's a mouthful.
I know, it is a bit of a mouthful, but it's a really neat technique.
Okay.
It lets us focus specifically on events that are happening at the cell surface,
Mm.
like watching molecules interact right at that boundary between the cell and its environment.
So like observing the cell's skin,
Yeah, exactly.
up close and personal.
Up close and personal, that's right.
Fascinating. It seems like there's No limit to how closely we can observe these tiny cellular worlds.
It really is amazing.
But is there a limit to how small we can see?
Oh, that's a great question.
It seems like we're already peering into this incredibly microscopic realm.
We are.
Can we zoom in even further?
The answer is yes.
Wow.
Scientists are always pushing the boundaries of what's possible.
Of course.
And we have a category of techniques called super resolution microscopy.
Super resolution.
Yeah.
So like Superman's vision for cells.
Haha, yeah, kinda.
Okay.
Super resolution microscopy allows us to break the limits of traditional light microscopy
What?
and see even tinier details within living cells. We're talking about structures on the nanoscale.
Wow, that's smaller than the wavelength of light.
It is.
How is that even possible?
It's complicated, but these techniques use these clever tricks with fluorescent molecules and advanced imaging processing to create these amazingly detailed images far beyond what was previously thought possible.
So we're not just watching cells move and divide anymore.
No.
We're seeing the individual molecules within them interacting.
That's right. It's opening up this whole new world of understanding about how cells work at the most fundamental level.
This is all so fascinating.
It is.
But what does it all mean?
Right.
What are some of the big picture applications of live cell imaging? How is it actually helping us understand the world around us?
Well, one area where live cell imaging is making a huge impact is in our understanding of cell migration, those microscopic chases we talked about.
Right.
By watching how cells move in real time, we can learn so much about how they respond to signals,
Okay.
how they navigate their environment, and how they interact with other cells and structures.
And why is that important? What does understanding cell migration actually help us do?
Well, it's crucial for understanding a wide range of biological processes.
Okay.
For example, wound healing
Hey.
relies on cells migrating to the site of an injury to repair the damage.
Okay.
On the flip side, understanding how cancer cells migrate is essential for developing strategies to prevent the spread of tumors.
So studying cell migration could lead to faster healing.
Yeah.
And better cancer treatments.
Exactly.
Oh.
And live cell imaging isn't just helping us understand movement, it's also providing these incredible insights into another fundamental process, cell division.
Right.
Remember that intricate dance of chromosomes we talked about earlier?
Of course, it was mesmerizing to watch.
With live cell imaging, we can watch that entire process unfold in real time,
Wow.
including all the steps of mitosis, where a single cell divides into two identical daughter cells.
And what are we learning from watching this cellular dance up close?
Well, we're gaining a much deeper understanding of the control mechanisms that ensure accurate cell division, which is essential for growth, development, and tissue repair.
Okay.
We're also learning what happens when these controls go wrong, which can lead to diseases like cancer.
So by watching cells divide, we can potentially identify new targets for cancer therapies.
That's the hope.
Wow.
And there's another critical area where live cell imaging is shining a light. Signal transduction.
Signal transduction. Remind me what that is again?
It's essentially how cells communicate with each other
All right.
and with their environment.
Right.
They're constantly sending and receiving signals, telling each other to grow, divide, differentiate, or even die.
So it's like the language of cells.
Yes, exactly.
Okay.
And with live cell imaging, we can actually eavesdrop on these cellular conversations.
Really?
We use these special fluorescent sensors that light up when specific signaling pathways are activated.
Oh, wow.
It's like having these tiny little spies inside the cell reporting back on what's going on.
That's so cool.
It is pretty cool.
So we can see how cells respond to signals from their environment, like hormones or growth factors.
Exactly. This is helping us understand a vast array of processes, from how the immune system fights off infections to how the brain forms new connections.
It's mind-boggling to think that we can now observe these intricate cellular conversations in real time.
It really is.
It's like we've been given a key to unlock the secrets of life itself.
It's a powerful tool.
It is, but I'm sure there are challenges, right?
It can't be easy to keep these tiny cells happy and healthy
Under a microscope.
Right.
While we're bombarding them with light
Yeah.
and trying to spy on their every move.
It's a delicate balance.
So what are some of those challenges?
One of the biggest is phototoxicity.
Phototoxicity, that sounds a bit ominous.
It basically means that exposing cells to too much light can damage them or alter their behavior.
So it's like we're trying to observe these delicate creatures without disturbing their natural habitat.
Precisely. It's a delicate balance.
Right.
Researchers have to carefully control the intensity and duration of light exposure
Okay.
to minimize any potential harm to the cells.
So how do they manage that? What are some of the strategies they use?
Well, one common approach is to use very sensitive cameras that can detect even faint signals, allowing us to use lower light levels.
Makes sense?
We also use special filters that block out harmful wavelengths of light.
Hmm.
And of course, we try to minimize the overall exposure time.
So it's all about finding the right balance between getting a good image and keeping the cells happy.
Exactly.
Got it.
And as technology continues to improve,
Right.
we're getting better and better at doing that.
That's good.
Another challenge is dealing with the sheer volume of data.
Oh, the data deluge.
Yeah, the data deluge.
That's a problem in so many areas of science these days,
It really is.
especially with all this new technology.
Exactly. And live cell imaging experiments can generate terabytes of data.
Wow, terabytes.
And analyzing all that information can be incredibly time-consuming and complex.
Right.
We need these sophisticated computational tools and algorithms to help us make sense of it all.
So it's not just about capturing the images, it's about processing and interpreting them.
Exactly. The data analysis is a critical part of the process.
Okay.
And then, of course, there's the ongoing challenge of resolution.
We talked about super resolution microscopy earlier.
We did.
Isn't that already pushing the limits of what we can see?
It is, but scientists are never satisfied.
Right.
We always want to see things in even greater detail.
Of course.
So researchers are constantly working on developing new techniques and technologies to push the boundaries of resolution even further.
It's amazing to think that we've come so far in our ability to observe the microscopic world,
It is.
but there's still so much more to discover.
Oh, yeah.
What an exciting time to be in cell biology.
I couldn't agree more. It's truly a field that's constantly evolving and pushing the boundaries of knowledge.
Well, this has certainly been a deep dive.
It has.
I don't know about you, but my brain is officially full.
Mine too.
We've covered so much ground today, learning about the incredible power of live cell imaging, from time-lapse microscopy to super-resolution techniques.
Right.
And we've explored how these tools are helping us unlock the secrets of cell migration, division, and signal transduction.
Absolutely.
It's truly mind-blowing.
It is. It's remarkable how far we've come.
It really is.
And it's constantly evolving. There's always something new to discover.
It's all very impressive, but I'm curious, how are these techniques being used to tackle real-world challenges? How are these tools making a difference in areas like medicine and human health?
That's a great question, and it's one that researchers are really actively exploring right now.
Okay.
For instance, take those microscopic chases we talked about earlier.
Right?
Cell migration.
Yeah.
Scientists are using live cell imaging to study this process in incredible detail, particularly in the context of cancer metastasis.
Metastasis, that's when cancer spreads, right?
Exactly. It's when cancer cells break away from the original tumor and travel to other parts of the body, establishing new tumors in distant organs. It's a complex and often devastating process.
So how is live cell imaging helping us understand it?
Well, by observing cancer cells in real time, researchers can see how they interact with their environment,
Okay.
how they squeeze through those tiny blood vessels, and how they invade other tissues.
So we're literally seeing these cells on the move, figuring out their escape routes.
Precisely. And by understanding the mechanisms behind their movement, we can potentially develop new therapies to block their spread. It's like cutting off their escape routes before they even have a chance to get away.
That's fascinating. It's like learning the enemy's tactics to outmaneuver them. So what other applications are there?
Well, let's move from movement to another fundamental process, cell division.
Right.
Remember that intricate dance of chromosomes we talked about?
Of course, it was mesmerizing.
Well, live cell imaging is allowing us to study this process in unprecedented detail, particularly in the context of developmental biology.
Developmental biology, you mean how organisms develop from a single fertilized egg?
Exactly. Cell division is the engine of development, and by observing it in real time, we can gain a deeper understanding of how tissues and organs form,
Right.
how cells specialize into different types, and what happens when these processes go awry.
It's incredible to think that we're not just watching cells divide, we're essentially watching the very foundation of life being built.
It is truly awe-inspiring. And speaking of building blocks, let's dive into the world of immunology. Remember how we talked about cells communicating through signal transduction?
Yes, like they have their own secret language?
Exactly. Well, immune cells are masters of communication.
Right.
And they rely heavily on signal transduction to coordinate their attacks against invaders like bacteria and viruses.
So how is live cell imaging helping us understand this microscopic battleground?
Well, imagine this. We can actually observe immune cells recognizing pathogens,
Wow.
activating each other,
Okay.
and mounting a coordinated defense to neutralize the threats.
So we're basically watching the immune system's battle plan in action.
That's a great way to put it. It's like watching a well-choreographed military operation unfold in real time.
That's incredible.
It is, and this knowledge is helping us develop new vaccines and immunotherapies to enhance the body's natural defenses,
Oh, wow.
giving our immune systems an even bigger advantage in the fight against disease.
It seems like this technology is revolutionizing our understanding of how life works at a fundamental level.
It really is.
It's opening up so many new possibilities. Are there any areas where you think live cell imaging will have a particularly big impact in the future?
Well, one area I'm particularly excited about is the study of how cells interact with each other in these complex tissues and organs.
Yeah.
You see, up until now, a lot of research has focused on individual cells in isolation.
Right.
But in reality, our bodies are made up of trillions of cells, all working together in this beautifully orchestrated symphony of life.
It's just like moving from observing individual instruments to appreciating the full orchestra.
Exactly. And with live cell imaging, we can begin to decipher these complex communication networks that govern tissue development, function, and repair.
Okay.
We can see how cells interact in real time, how they influence each other's behavior, and how they work together to create the amazing complexity of a living organism.
That's incredible. It feels like we're on the verge of a whole new era of understanding in biology.
I think so too.
But what about the brain? It's such a complex and mysterious organ. Can live cell imaging help us unlock its secrets?
Absolutely. Neuroscientists are already using live cell imaging to study how neurons connect and communicate with each other,
Okay.
how they form new synapses,
Right.
and how they respond to stimuli.
So we can actually watch thoughts forming in real time.
Well, not quite. Thoughts are incredibly complex phenomena
Yeah.
involving vast networks of neurons firing in these intricate patterns.
Yeah.
But live cell imaging is giving us this glimpse into the fundamental processes that underlie those patterns, how individual neurons behave, and how they connect to form the circuits that make our brains work.
It's still pretty amazing. Imagine being able to see how learning and memory physically manifest in the brain. It's like watching the brain learn.
It really is remarkable. And as imaging technology continues to improve, who knows what other wonders of the brain we'll be able to witness.
Yeah.
Perhaps one day we'll be able to see those complex thought patterns you mentioned.
That would be mind-blowing. But with all this incredible potential,
Yeah.
are there any challenges or limitations we need to consider? It can't be easy to keep these tiny cells happy and healthy under a microscope, especially when we're trying to observe them over long periods.
You're right. There are definitely challenges. For one thing, cells are delicate.
Right.
And the very act of observing them can sometimes disrupt their natural behavior. So we have to be very careful to minimize any potential harm caused by the imaging process itself.
So it's like trying to observe a fragile ecosystem without disturbing its delicate balance.
Exactly. And that's not all. Live cell imaging experiments often generate massive amounts of data.
Right, the data deluge.
Yeah, we're talking about terabytes of information that need to be processed, analyzed, and interpreted.
Wow.
This requires powerful computers and sophisticated software, not to mention skilled researchers who can make sense of it all.
It sounds like big data meets biology.
It does, it does.
But despite these challenges, the future of live cell imaging seems incredibly bright. What are you most excited about in the years to come? What are the big unanswered questions that you think this technology might help us address?
Oh, there are so many exciting possibilities. One area that really fascinates me is the potential to use live cell imaging to study how cells respond to drugs in real time.
So we could actually see how a drug interacts with its target inside a living cell.
Exactly. Imagine being able to watch how a cancer drug targets and destroys tumor cells.
Wow.
Or how an antibiotic kills bacteria.
That's incredible.
This could revolutionize drug development,
Right.
allowing us to design more effective and targeted therapies with fewer side effects.
That would be a game changer. It seems we're really just at the beginning of this incredible journey of discovery.
I think we are.
What other areas hold promise for live cell imaging?
Well, another area that's really intriguing is the potential to use this technology to study how cells age.
So we could actually watch the aging process unfold at the cellular level.
Precisely. By observing cells over time, we could gain a much deeper understanding of the molecular mechanisms that drive aging.
Wow.
and perhaps even identify ways to slow down or reverse the process.
Wow, that would be revolutionary.
It would.
The implications for human health and longevity are staggering. But before we get too carried away with the future, let's bring it back to the present.
Okay.
What are some of the key takeaways you'd like our listeners to remember about live cell imaging?
I think the most important thing is that this technology is allowing us to see life in a way it was never before possible.
Hmm.
We're no longer limited to those static snapshots of cells. We can now observe them in action, see how they behave, how they interact, and how they respond to their environment.
Right.
This is giving us unprecedented insights into the fundamental processes that govern life, from the very beginnings of development to the complex workings of the brain and the immune system.
It's a reminder that even the tiniest things can hold the greatest secrets.
They really can.
And with tools like live cell imaging, we're finally starting to unravel those secrets and gain a deeper appreciation for the incredible beauty and complexity of life itself. And it's really amazing to think about how far we've come in just a few decades.
It is.
What started as these static images
Right.
has evolved into these incredible dynamic glimpses into the world of living cells.
It's a testament to human ingenuity and our pursuit of knowledge.
It really is.
And the best part is we're still just scratching the surface.
Exactly. There's so much more to explore. So what are some of the big unanswered questions that you're most excited about? What mysteries are you hoping live cell imaging will help us solve in the coming years?
Well, one area that I'm particularly fascinated by is the study of how cells interact in these complex tissues and organs.
Okay.
You see, so far a lot of the research has focused on these individual cells in isolation.
Right.
But in reality, our bodies are made up of trillions of cells,
Yeah.
all working together in this beautifully intricate way.
So it's like we've been studying the individual pieces of a puzzle.
Yeah.
But now we're finally starting to see how they all fit together to create the bigger picture.
Exactly. And with live cell imaging, we can begin to understand these complex communication networks that govern tissue development, function, and repair.
Okay.
We can see how cells influence each other's behavior, how they cooperate to perform tasks, and how they respond to changes in their environment.
It's like we're moving from a solo performance to a grand symphony, where each cell plays its part in creating this harmonious whole.
A perfect analogy.
And speaking of complex systems, what about the brain?
Oh, the brain.
It's still such a mysterious organ. Can live cell imaging help us unlock its secrets?
Absolutely. Neuroscientists are already using this technology to study how neurons connect and communicate,
Okay.
how they form new synapses, and how they respond to various stimuli.
So we can actually watch the brain learning in real time.
Well, not quite in the way we typically think about learning.
Right.
Remember, thoughts and memories are these incredibly complex phenomena that involve vast networks of neurons firing in these intricate patterns.
Yeah, it's a lot going on in there.
It is, but live cell imaging allows us to see the individual neurons, how they behave, and how they connect to form those networks.
Right.
It's like seeing the individual wires and circuits that make up a complex computer.
So it's still pretty amazing.
It is.
Imagine being able to see how those connections change as we learn new things or form new memories.
It's truly remarkable, and as imaging technology continues to improve, who knows what other wonders of the brain we'll be able to witness. Perhaps one day we'll be able to see those complex thought patterns you mentioned, or even understand how consciousness arises from this intricate dance of neurons.
That would be incredible, but for now, even just being able to observe these individual neurons and their connections is a huge leap forward.
It is.
It's giving us a whole new level of insight into the brain and how it works.
Right, and it's opening up these exciting new possibilities for understanding and treating neurological disorders.
Oh.
Imagine being able to see how Alzheimer's disease disrupts neuronal connections, or how Parkinson's disease affects the activity of specific brain cells. This knowledge could lead to new therapies that target the root causes of these diseases, rather than just treating their symptoms.
That's amazing to think about the potential impact on human health.
It is.
It feels like we're on the cusp of a new era of discovery in neuroscience.
I think so too.
Thanks in no small part to these advances in live cell imaging.
Absolutely.
It's incredible.
It is a testament to the power of this technology.
It is.
And its ability to shed light on these hidden workings of life itself, and it seems like we've only just begun to explore the possibilities.
Exactly. And as we continue to push the boundaries of what's possible with live cell imaging, who knows what amazing discoveries await us.
It's an exciting time.
It really is. It's been a fascinating journey exploring this world of live cell imaging with you. Thanks for sharing your expertise and your insights with us.
It's been my pleasure, and to all our listeners out there, remember, stay curious, keep exploring, and never underestimate the power of a microscope.